Ultrasound pulses to brain send mice into a hibernation-like state. Experiments offer an intriguing hint at technology that could induce torpor in humans in the future. For many animals, life is a cycle of scarcity as well as plenty. Hibernating creatures curl up underground in winter, slowing their metabolism so they can make it to spring without food. Even laboratory mice, if deprived of food, can enter a state called torpor, a kind of standby mode that economizes energy. It's something humans have long fantasized about for ourselves. If we ever leave this planet and travel through space, we will experience our own time of scarcity. Science fiction writers tend to imagine a mysterious technology that keeps humans in stasis, able to survive centuries of silence before emerging into a new life. For now, it's a technology that's out of reach. But as scientists work to understand states like torpor and hibernation, tantalizing details about how the brain controls metabolism have emerged. Researchers reported in the journal Nature Metabolism on Thursday that they've been able to send mice into a torpor-like state by targeting a specific part of the brain with short bursts of ultrasound. It's unclear exactly why ultrasound has this effect, but the findings suggest that studying the neural circuits involved in torpor could reveal ways to manipulate metabolism beyond the lab. Ultrasound devices, which generate high-frequency sound waves, are best known for their imaging powers. But they have also been used by neuroscientists to stimulate neurons. Correctly tuned, the sound waves can travel deep into the brain, said Hong Chen, a professor of biomedical engineering at Washington University in St. Louis and an author of the new paper. In 2014, William Tyler, now at the University of Alabama, Birmingham, and his colleagues applied ultrasound to a sensory region in the brain and found that it enhanced a subject's sense of touch. A growing body of work is exploring ultrasound as a treatment for disorders like depression and anxiety. Curious about a brain region that regulates body temperature in rodents, Dr. Chen and her colleagues constructed tiny ultrasound mouse caps. The devices trained 6 bursts, each consisting of 10 seconds of ultrasound, on the selected area of the rodent's brain. Researchers who study the brain with ultrasound must tune their devices carefully to avoid heat that can damage tissues. The mice, the researchers noticed, stopped moving. Measurements of their body temperature, heart rate and metabolism showed a pronounced dip. The mice stayed in this state for about an hour after the ultrasound bursts and then returned to normal. Looking closer at neurons involved in this response, the researchers identified a protein in their brain membranes, DRPM2, that appears to be sensitive to ultrasound. When the researchers reduced levels of the protein in mice, the mice became resistant to ultrasound's effects. That's an important step toward understanding how ultrasound affects neurons, said Davide Faloni, a researcher at the Icahn School of Medicine at Mount Sinai in New York City, who studies the brain using ultrasound. The details have largely been elusive. But it's also possible that heat generated by the ultrasound, and not just the ultrasound itself, is affecting TRPM2 in the brains of mice, a point that was raised by Masashi Yanagisawa and Takeshi Sakurai of the University of Tsukuba in Japan, in separate interviews. The two have studied neurons in this brain area, and their connection to states of torpor. Both may be in play. Can I get around a no-pet policy with an emotional support animal? In one of the most tantalizing parts of the study, the researchers checked to see whether animals that don't typically experience torpor, rats, behaved differently when the brain region was stimulated with ultrasound. Indeed, they seemed to slow down, and their body temperatures dropped. We have to be careful with the rat data, Dr. Chen cautions. So far, they only have information about temperature, not metabolic rate and other factors. Could ultrasound be a way to change the metabolism of larger animals with no history of torpor, like humans? It's an intriguing idea, Dr. Sakurai said. At this stage, he said, it remains an unanswered question. Aiming ultrasound at the brain raises hope of new treatments. Approach could lead to new ways to treat epilepsy, depression, and Alzheimer's disease. As a way to see inside the body, revealing a tumor or a fetus, ultrasound is tried and true. But neuroscientists have a newer ambition for the technology, tinkering with the brain. At frequencies lower than those of a sonogram but still beyond the range of human hearing, ultrasound can penetrate the skull and boost or suppress brain activity. If researchers can prove that ultrasound safely and predictably changes human brain function, it could become a powerful, non-invasive research tool and a new means of treating brain disorders. How ultrasound works on the brain remains mysterious, but recent experiments have offered reassurance about safety, and small studies hint at meaningful effects in humans, dampening pain, for example, or subtly enhancing perception. "I've seen a lot of tantalizing data," says Mark Cohen, a neuroscientist at the University of California, Los Angeles, UCLA. "While the challenges are very large, the potential of this thing is so much larger that we really have to pursue it. Scientists can already modulate the brain non-invasively by delivering electric current or magnetic pulses across the skull." The U.S. Food and Drug Administration (FDA) has approved transcranial magnetic stimulation (DMS) to treat depression, migraine pain, and obsessive compulsive disorder (OCD). But unlike magnetic or electric fields, sound waves can be focused like light through a magnifying glass on a point deep in the brain without affecting shallower tissue. For now, that combination of depth and focus is possible only with a surgically implanted wire. But ultrasound could temporarily disrupt a deep human brain region, the almond-shaped amygdala, a driver of emotional responses, for example, or the thalamus, a relay station for pain and regulator of alertness to test its function or treat disease. Results in animals are encouraging. Experiments in the 1950s first showed ultrasound waves could suppress neural activity in a visual region of the cat brain. In rodents, aiming ultrasound at motor regions has triggered movements such as a twitch of a paw or whisker, and focusing it on a frontal region of monkey brains can change how the animals perform eye movement tasks. But it's technically tricky to aim ultrasound through thick, dense skull bone and to show its energy has landed at the intended point. And ultrasound's effects on the brain can be hard to predict. How much it boosts or suppresses neural activity depends on many parameters, including the timing and intensity of ultrasound pulses and even characteristics of the targeted neurons themselves. "I have tremendous excitement about the potential," says Sarah Hollingsworth-Lisenby, a psychiatrist at the National Institute of Mental Health who studies non-invasive neuromodulation. "We also need to acknowledge that there's a lot we have to learn," she says. For one thing, researchers are largely in the dark about how sound waves and brain cells interact. "That's the million-dollar question in this field," says Mikhail Shapiro, a biochemical engineer at the California Institute of Technology. "At high intensities, ultrasound can heat up and kill brain cells, a feature neurosurgeons have exploited to burn away sections of brain responsible for tremors. Even at intensities that don't significantly increase temperature, ultrasound exerts a mechanical force on cells. Some studies suggest this force alters ion channels on neurons, changing the cell's likelihood of firing a signal to neighbors. If ultrasound works primarily via ion channels, that's great news," Shapiro says, because that means we can look at where those channels are expressed and make some predictions about what cell types will be excited. In a preprint on BioRxiv last month, Shapiro's team reported that exposing mouse neurons in a dish to ultrasound opens a particular set of calcium ion channels to render certain cells more excitable. But these channels alone won't explain ultrasound's effects, says Seung-Shik Yu, a neuroscientist at Harvard University. He notes that ultrasound also appears to affect receptors on non-neuronal brain cells called glia. "It's very hard to develop any unifying theory about the exact mechanism of ultrasound," he says. Regardless of mechanism, ultrasound is starting to show clear, if subtle, effects in humans. In 2014, a team at Virginia Polytechnic Institute and State University showed focused ultrasound could increase electrical activity in a sensory processing region of the human brain and improve participants' ability to discern the number of points being touched on their fingers. Neurologist Christopher Butler at the University of Oxford and colleagues have tested ultrasound during a more complex sensory task, judging the motion of drifting, jiggling dots on a screen. Last month at the Cognitive Neuroscience Society's annual meeting online, he reported that stimulating a motion processing visual region called MT improved subjects' ability to judge which way the majority of the dots drifted. Ultrasound's effects have so far been subtler than those of DMS, says Mark George, a psychiatrist at the Medical University of South Carolina, who helped develop and refine that technology. "With DMS, you put it on your head and turn it on and your thumb moves," he says. But the ultrasound experiments that prompted paw twitches in mice used intensities so much higher than what we are being allowed to use in humans. Regulators have. Christopher Butler at the University of Oxford and colleagues have tested ultrasound during a more complex sensory task, judging the motion of drifting, jiggling dots on a screen. Last month at the Cognitive Neuroscience Society's annual meeting online, he reported that stimulating a motion processing visual region called MT improved subjects' ability to judge which way the majority of the dots drifted. Ultrasound's effects have so far been subtler than those of TMS, says Mark George, a psychiatrist at the Medical University of South Carolina, who helped develop and refine that technology. With TMS, you